slide for that.
Oh, yay.
So first of all, I only talk about a very, very small snippet. It's kind of a teaser, actually. So if you want to ask a question, I've got some other slides. But we've had various funding, mostly from the NIH, but also the tobacco cure funds. I thought the calcium phosphate silicate was such a swell idea. Back in 2005, I started a company with two other colleagues, Keystone Nano. And I'm happy to say we're in our first clinical trial right now. Started about 1.5 years ago. And it's going very well, actually. We're actually helping some patients who came to us with literally no hope at all. So I was a little nervous about giving this talk, to the extent I've literally never given a talk on the microbiome. I've become quite familiar with what's called the tumor microenvironment, or the TME. You'll see that pop up in some of the slides. So I put together some questions. And then the meat of the presentation is hopefully some answers or some things that make the questions a little bit more visible. And then inevitably at the end, I had to do this interconnectedness of all. Because in cancer treatment, as you'll see, I'm experienced on both ends. I'm a stage 3 cancer survivor. My wife is a breast cancer survivor, and that all happened in the middle of all this development stuff that we were doing. So it's both ironic or funny if you really have a dark sense of humor, and luckily I do. So I grew up in Florida, but I'm actually a Hoosier by birth. Does anyone know what the Hoosier state is? Yeah, Indiana, right in the heart of the country. Flat as Florida, which is why I wound up here in beautiful State College with all the mountains. I got my PhD and my BS both in material science, but I moved across campus. I was bartending, and someone came in and started talking about biomaterials, and I said, I think I want to check that out, and here I am. I did my PhD on kidney stone disease. I did my Fulbright postdoc on kidney stone disease, pathological biomineralization. I think 45 years later, I think we've actually figured it out now. That's something else. From my postdoc, I went into kind of a think tank, Patel Memorial Institute, solve problems for industry. It was great for a young scientist to go to, because I learned about putting together proposals, what we call marketing in industry, but we call grantsmanship in universities, and how to manage also. So that was terrific. I've been a professor of material science and engineering since 2002, and then a few years ago, biomedical engineering, where I have strong connections in pharmacology down at Hershey, asked me if I wanted a courtesy appointment. I'm director of the Penn State Center for Nanomedicine and Materials as well. Well, personally, I'm a husband, actually twice over. I learn on the job. A father, an uncle, a grandfather, 2 times over, a brother and a son, like many of you, unless you happen to be of the other gender, in which case you're not a husband, probably. I'm a stage 3 cancer survivor. Oh yeah, and heart disease. And for the young people, so I thought the heart disease was going to kill me. So at 50, I did not get that colonoscopy. I blew that call. 56, I got it, stage 3 cancer. So get that colonoscopy. If it's in your family, get it by 40. And the most difficult job I've ever had, not because my wife is difficult, she is. She's a Irish girl, Irish Catholic girl from Philadelphia. She challenges me constantly. And this marriage has now lasted 30 years. That was the hardest thing I've ever done. So I come at it both as an advocate and as a research scientist. And it's a little bit different perspective than you're probably going to get from other people. So when I started to put this talk together 2 or 3 weeks ago, I thought, okay, I've been collecting papers for the last 6 or 7 months because Mary Lee had me scheduled last semester, and I kind of blew her off because I was, again, they had a pretty heavy teaching load. And so I came up with these 4, but when I started to really think about it deeply in terms of how to present it in a logical flow, and of course, as a cancer survivor, why wouldn't I come up with, do cancer treatments affect the microbiome? First, because believe me, as a cancer survivor, I know they do, terribly in some ways. So I rearranged this, and I'm very proud of this animation. Does cancer affect the microbiome? Can the microbiome be used to detect cancer? Do cancer treatments affect the microbiome? And does the microbiome affect, I think I asked that, no, I guess not. Does the microbiome affect the cancer treatment? And so I'm going to give you everything you need to know from this talk. Does cancer affect the microbiome? Yes, it does. Can the microbiome be used to detect cancer? Yes, it can. Do cancer treatments affect the microbiome? Yes. Actually, hell yes. Does a microbiome affect the cancer treatment? And that's a yes. So you have everything you need to know, and you can leave now. Well, and going through, this has been a wonderful exercise because I'm really out of my comfort zone. And I looked up, and thank God for Wikipedia and Google, and I looked up all kinds of new words to me. But this one I knew. I think of cancer as a parasite. And I think of cancer survival in terms of functional significance. So what we're finding out is cancer usually is created by stem cell differentiation gone awry. We know that stem cells generate cells that don't survive, don't serve a function, and they get removed rather quickly. But cancer seems to have at least 3 characteristic survival traits. It has all these others, and I'll be talking about some of these in a little bit more detail. Unlimited multiplication. The governor that controls cellular division up to a certain point, mostly around DNA damage, where it will stop and the cell will go into apoptotic cascade, cancer doesn't have. It tolerates hugely damaged amounts of DNA, for example. Unlimited multiplication. It also escapes from growth suppressors. Actually, it does more than that. It actually hijacks their function. It maintains proliferation signaling, and it evades immune destruction as well. So those are some pretty good survival traits if you're trying to survive. Unfortunately, the host is badly affected. I'm going to very quickly give you a little bit, you probably all know it, but the hematopoiesis, stem cell differentiation, the formation of a few of the blood cells, and how cancer evades the destruction by hijacking the immune system. And you'll see that popping up in some of the others. So cancer signals the differentiation to create immunosuppressor cells through a host of distorted, mostly distorted cytokines. So there are T cells that you'll see, but not healthy T cells. These T cells actually foster the growth of the cancer. B cells get hijacked too. In other words, this pluripotent cell does not differentiate into the immune effector. It differentiates into the immune suppressor cells. And in fact, someone with stage 3 or 4 cancer will often have an immune system that's compromised by the cancer. I like to say, so of course, the chemotherapy philosophy developed really in the late 50s and 60s to knock down the rapidly dividing cells, further tanks the stem cell differentiation to immune cells. And neutropenia is something that you often hear about. That means you have a compromised immune system. And they often have a compromised immune system going into the treatment with usually and often bad outcomes, unfortunately. We've discovered something back in through 2008 through 2013, and now we're moving it further toward the clinic, that we called photoimmunonanotherapy. which blocks the cancer signaling and resurrects the immune system. Now, the reason I gave you this is we're going to talk about one recently evaluated immune therapy in human trials. And sadly, I would have to say it failed in terms of that patient cohort. 40% of the patients were helped, 60% were not. And it was directly attributed to the microbiome that those host cancer patients had. Interesting. Okay, so you can look at this and understand it completely. I thought I'd put this in, but I'm very careful, and Carol Lee and I were talking about getting these papers up so you can take a look. Now, embedded in the small print, and I'll elaborate on this, are how T cells will get upregulated in the presence of the signaling from the cancer to T cells that suppress the immune system. And B cells will get upregulated that will suppress the immune system. Often when you see T cells associated with a tumor, and here's a test for you. What's the minimum size tumor that we can routinely detect? What size? 2 millimeters maybe? Nah, at least a centimeter. And by the time you hit a centimeter, it's usually vascularized, which means it is now systemic. It's got a blood supply, it's sending the signaling agents. So one of the things we're working on is much earlier detection of cancerous lesions and where they are. But in any case, so a bunch of different things occur when you have a tumor. One of the things that occurs, and this was relatively hard to ferret out, is something close to my heart as an inorganic chemist originally. I guess I'm a materials chemist now because I've been doing that for 40 years. And that is that if you have a blood vessel, the blood vessels that the cancer forces the body to form, because the cancer also sends out angiogenic signaling, are usually formed so fast they're leaky. And so they'll have much larger defects than a normal blood vessel, where you can have the leaky vasculature will deliver particularly nanoparticles across through the extravasation. And so you can treat tumors even if you don't have a target, not as well as you can with a target. But if you have a tumor in close proximity, and you always do because the tumor demands that and grows the blood vessels, the cancer cells that are nearest to the blood vessel get the full milieu of nutrients and oxygen. But their metabolic rate is so high that the cancer cells away from the blood vessel don't get much oxygen. And this hypoxia leads to a decrease in pH. So and this is obviously measured from a small lesion. Our experience with centimeter scale or larger tumors that are vascularized is you could actually get into pH 4 or 5 regime. And I didn't find any papers that looked at the microbiome when that occurs. That would be interesting to look at. So you have a leaky vasculature, you've got extreme in pH. There are almost always inflammatory responses going on around tumors. And if the inflammatory processes are making some movement toward trying to destroy the tumor, you'll see necrotic lesions, and you'll have a higher pH from the necrosis. But most tumors are pretty acidic. So my question to you, how can these affect local flora? And I'm going to talk about how the remote flora can be affected as well. In other words, the gut microbiome. I know you can't read this. I thought this was a very cool study, but this was a large database study. And in this, for colorectal cancer, something near and dear to my own heart, that was a joke. Dark humor. Well, no, it's not near and dear because it's long gone. They showed, and this isn't the way they came up with really good prediction. They also looked at bin data, massively bin data, and they're hitting the 90%, 80 to 90% positive mark, and very low negative results. So they showed that you can establish adenoma, carcinoma, and that suggests that we can detect cancer. And in fact, that little box guy, I always thought that was a scam, to be honest. But the FDA approved, I think it says 2012 or 2013, approved this in 2012, where you literally send your stool sample in, and they will take a look at the microbiome and tell you if your risk for colorectal cancer. How cool is that? Now, why can they do that? Well, one of the few things the FDA does not regulate are body fluids. And of course, stool is a body fluid, urine's a body fluid, but urine is particularly singularly uninteresting because it's sterile. You don't get any real markers. You might get some protein markers for pancreatic cancer, for example. But the stool is rich and a pretty fair indication of what the heck's going on. Okay, so do cancer treatments affect the microbiome? I think I expressed my personal opinion from my own experience. Yeah, sure. Going back?
Yeah, go back one. So right there. So colonoscopies are horrendous. I mean, the lead up to
I've had probably 20 or 30.
I'm glad
I agree.
I'm glad you enjoy them so much, but they're really for most of us, they're not what we would look forward to. Picking up a stool sample seems to be, even though that might be considered dégueulasse.
Not to a colon cancer survivor.
Exactly. So I'm just curious. Would you at this point use that instead of colonoscopy?
No.
Okay.
However, I will use this. In fact, in my studies, I came across a group, it may be covered by insurance, but I doubt it, but I'm going to pay the $500, that will look at my microbiome over the course of the next 6 months. I got my first 2 kits yesterday in the mail. And so I'm curious, what's my microbiome look like? I have no idea, to be honest.
Daryl would also like this.
Well,
I have more than enough shit for you, Daryl. That's colon cancer humor.
Yeah.
But no, it's not regulated. So people are starting to take charge of their own health, as you should. So, for example, the first indication I had that my microbiome was fouled up, I took 12 rounds of chemotherapy. I was stage 3 after all. Every 2 weeks for 6 months, I got thrush. I got a yeast infection in my throat. And that was a pretty fair indication that between the surgery for colon, they really wipe out your gut microbiome with heavy antibiotics, and the chemo then finishes the job in your bloodstream and elsewhere. So you lose most of the bacteria. I'll show you what the result of that is. And so there's no ambiguity. This graphic, this is the microbiome in a host of patients before. This is the microbiome after. So there's no ambiguity. These are the populations, and the green is the healthy microbiome, and of a large pool of patients. And this is the chemotherapy microbiome. You lose a lot of bacteria, and the diversity increases tremendously. When I was in the hospital right after my surgery, there was a Clostridium diff that went through the patients, and they put me on Flagyl just to make sure I was already completely antibiotic up, but the Flagyl is specifically for C. diff. And actually, I was allergic. So I wound up being in the hospital 10 days. It's not the bad part. Actually, there was a good part. I lost like 20 pounds because I was on a water diet. Do you know what they serve you? Water. Jell-O was the filet mignon of my meals over 10 days. So if you want to lose weight, get colon cancer. Okay, I thought this was I gained it back. I lost 25 pounds, I gained back 20. So inevitably, that 5 pounds was the 12 inches of large intestine they took out. I love this figure. Will this follow me? Oh. That's okay. I'll try to use the pointer. Oh, can you see that?
Yeah.
Okay. There should be a pointer someplace. Okay, but in any case, so do cancer treatments affect the microbiome? The most consistent part of this is here. The rest of this is where the microbiome affects the cancer treatment. And I'll come to that slide in a few minutes. So you get translocation. This is a pretty grim thing. With at least 2 of the chemotherapies, if they are cleared through the liver, hepatobiliary tract, and go into the small intestine, in the small intestine, and this is cyclophosphamide is one of them, and I'm not sure I can read the other one, but I'm familiar with both. Oh, doxorubicin. That's used for breast cancer. It's a second tier now, but it's used for breast cancer. So is this like cyclophosphamide as kind of a second chemo. The microvilli shorten up. They get damaged by the chemo going through the gut. And the second thing that happens is the mucosal membrane gets compromised. In other words, it no longer is the barrier to things like, well, the bacteria that are in your gut. And so I wonder that they've found the bacteria in the lymph system of the host. So that releases the bacteria. And of course, bacteria themselves can be pathological. But what's really dangerous about bacteria, if they proliferate, is that they die, of course, they'll produce large amounts of endotoxin, and there's nothing medically that you can do other than transplant their blood. Well, the problem with that is now it's in the lymph system, and they're proliferating. So this is a very, very, very bad side effect. I know you can't read this. I gave you the reference. I'll give you the paper. But there's all kinds of connections between specific chemotherapeutics. There's only a few in this where they haven't identified the microorganism. Oxaliplatin, I was on that one. Not a good kind. Mediated by TLR and reactive oxygen species production by myeloid cells. Well, that's not a good thing. So that's not known. Here's another one, CPT11, irinotecan, and we'll talk about that in a few minutes. Not known, etc. So we're starting to get a handle, but there's a lot of bacteria in the human body, and tracking down them. Here we are at this was 2017, last year when this was published. I hope that's not the full body of knowledge, but this is the kind of data that the NIH should be collecting. Really collecting and creating a database. And I don't know if they have. I didn't contact anyone at the NIH at this point. I didn't know to, I have to admit. Well, what does the chemotherapy do to the cancer treatment? Well, in the case of a recently vetted immunomodulation trial on humans, of course, clinical trial, it can lead to 60% of the patients not being affected at all. And that's bad if you're a cancer patient. And only 40%, depending on which bacteria are at the higher populations. That's really scary. But it also means that we could, a lot of times when we think about drug resistance in chemo, it may not be the chemo. It may not be the cancer. It might be the microbiome. And I'll offer some of the solutions. So the immunomodulation, there was one recent, I think there's a typo, PD-1 immunomodulator for cancer. The bifidobacterium modifies the tumor-specific T cell induction and increases the T cell numbers in the tumor microenvironment. That's a good thing. The Bacteroides, did I pronounce that right? Actually blocks the T cells so they can't do their job. So what's happening is the immune therapy is creating T cells. that are doing their function as immune cells and destroying the foreign tissue that is the cancer. The microbiome can block that. Yeah.
I have a question. So how are these chemotherapies normally delivered? Is it orally, or is it IV, or a mixture?
I don't know of any first-line chemotherapies that are given orally. They're given, first line are the most toxic. So they're given by IV. And usually, like I had a subcutaneous port put in. And so I got all of my chemo through that. My wife had terrible side effects in terms of getting a systemic bacterium from the surgery. And so she had a PICC line put in. That's a silicon rubber. And very fortunately, she had 4 rounds of chemo every 3 weeks. That was docetaxel and cyclophosphamide, actually. And she's got pancreas problems. No shock there. And that actually lasted. They were worried. The chemotherapy, the free drug, is so aggressive it will often dissolve the lines. But the silicone rubber held up well. So she didn't have to go through the little minor surgery, any more minor surgery. She did have 15 major surgeries over about 7 or 8 years. The metabolism and enzymatic degradation is also rather insidious. Irinotecan, I'm most familiar with it because now it's been a protocol of irinotecan with oxaliplatin and 5-fluorouracil has been recommended for pancreatic cancer. Not accepted as a protocol, recommended. There is no state of care for pancreatic cancer. And what they found with irinotecan is that a direct bacterial modification of basically of the drug that turns it into a completely toxic substance. The bacteria turns it into it's already toxic, right? But it turns it into a more toxic substance, and this causes diarrhea and colitis in the patient. Do you understand why cancer patients often have diarrhea? I didn't. Well, so they're targeting they're targeting the rapidly dividing cells, which of course includes all the stem cell and their differentiation. And of course, the lining of your stomach turns over every 24 hours. That we don't have anything in our body that can really last in the pH 2 that you have at times in your stomach. And of course, your stomach, I like to think of this pouch as mesenchymal tissue, but probably isn't. But in your stomach, you have mesenchymal tissue surrounding everything. So these are stem cells ready to differentiate into the endothelium and restore the lining of the stomach. When you're on chemo, that does not happen. So the lining of the stomach goes away and there's nothing replaces it. My wife and I did not have issues with nausea, so we were relatively lucky in that regard. But that's one of the reasons you get nausea. That's not so much diarrhea as nausea. Okay, again, I put this in because this will be up online, right?
Sure.
Okay. So I gave you this. I thought this is the immune therapy. One's for a type of melanoma, another one's for sarcoma cancer. And the key here are the red lines. All the red lines are when the immune therapy is delivered, excuse me, with an antibiotic present. Because a broadband antibiotic pretty much wipes out a lot of the microbiome in the gut. And as a result, the good bacteria, which I'll show you in the next slide, the good bacteria is wiped out and you lose the effectiveness. Now, you've got cancer patients. Their immune system's compromised. I had the flu, but of course there's nothing you do for flu, basically. But I had the flu during my chemotherapy because I just said, hell with it. I'm going to meet with my research group. I'm going to go out and watch my son swim, and to hell with everything else. My wife sequestered herself. She didn't get the flu. Oh, well. But I wonder how well the chemotherapy worked in the absence. So that's a microbiome change. And specifically, in this very nice paper, the It's nicely summarized. So bacteroideales, is that right? Oidales. If you have that, you've got a problem. This was about 60% of the patients in this trial. And you'll find that the prior paper was in a edition of Science that came out January 5th. So again, this is all fairly recent. There are a series of papers where they showed, holy mackerel, the microbiome affects this immune therapy. If you have, again, glasses on, Bifidobacterium akkermansia and fecal bacterium, it's going to work. Isn't that bizarre? It's effective treatment. Now, how effective is this immune therapy? When they talk about doing a bone marrow transplant for leukemia patients, what they're trying to do is restore an effective hematopoietic system into the patient, because the leukemia has knocked down the healthy hematopoietic system and really hijacked everything. So this is a really profound instance where not knowing the microbiome and not accounting for it led to failure in 60% of the patients. So does the microbiome affect cancer treatment? Well, absolutely. And these are various things that I already talked about. You get direct migration from this leaky villi. You can get migration, and this is referring to small molecules and bacterial cell walls that can lead to an endotoxin response. So T-cell plasticity changes, lots of migration, lots of T-cell migration, lots of effects. So this is the way that the microbiome in the gut can remotely affect the cancer treatment at a remote lesion. You know what this means for you guys? Opportunity. Seriously. Now, this was from that nice paper by Alexander, and I forgot, I'll make sure before I upload this, I put the citation. Because he was all about the immune system, but I suspect most chemotherapeutics will work much better if we take into account the microbiome. And so he's showing that you really have a triumvirate. One of the papers actually had that in the title, between the antigens, the metabolites, and the effector and suppressors. And all of these work together to either foster good treatment or to foster poor outcomes. I suspect many of the cancers that often have bad outcomes have probably grossly affected the microbiome. And so cancer patients don't have a normal, healthy microbiome. And then this last paper did a nice job of proposing, in all of these, they have a combination of antibiotics, but targeted antibiotics to kill specific types of bacteria. I don't know if we can do that effectively, but yeah, it's worth a shot, as well as probiotics. And we had a discussion in the committee, which probiotics can survive the gut? You know what I mean? They go to pH 2. Can we harden them up some way? I think we can, actually. So yeah, this person spun 4 different scenarios. Every one of these deals with personalized medicine. How personalized? How hard is it to get your microbiome evaluated? Doesn't seem like it's very hard, but more on that when I get my microbiome evaluated. Oh, yeah.
I'm curious, do you know anything about sort of how they develop chemotherapeutic treatments in the first place? Like, are these basically screened against cultured cells, or what's the process for sort of determining that a compound is
Well, some of the older chemos there was only one mammalian cell type as of about 1952, and that was the Henrietta lax cervical cancer cells. And those were deployed. She didn't know. She died a few months after I was born, actually. and she had an advanced stage of cervical cancer. But one of her caregivers, of course, they took biopsies, and they thought he had tried to culture the cells, and mammalian cells had not been successfully cultured as an immortal cell line. Now we can buy frozen cells, divide or thaw them, divide them up, and use them, and that's the way we do mammalian cell culture in vitro studies, or even for injection into athymic mice to study the effect of the local environment in an actual living host. So but I think Henrietta actually get a lot of credit, because for me personally, I got my first polio vaccine in 1955 at the ripe old age of 3. Us kids would line up. I guess I'm probably older than anyone here. We would line up at the local care center, and we would get our booster shots. And if you didn't like needles, well, that's just too bad. You're going to get your booster shot for polio. So when I was a kid, all the way through high school, in any classroom of 30, at least one kid was there that had polio. Have any of you seen any polio? That was arguably the greatest government health program in the history of the world, because polio virtually disappeared worldwide by about 1962, when the oral medication that was permanent was first deployed. So those cells were absolutely critical, because the investigator found he could freeze them, thaw them, and they were immortal. And they're HeLa cells, of course. So in 1952, there was another watershed called the discovery of the double helix by Watson and Crick. And some bright person, I haven't looked up the development, by about 1958 or 59, one bright person, I'm not going to pretend I control a molecule, one bright person took uracil and said, hey, we could put a fluoride on this. That was known for something in medicine. We could put a fluoride on this, I'll bet you it'll screw up RNA. So that's 5-fluorouracil. So that's been around, uracil. That's been around since 1960-ish. It doesn't just screw up RNA, it screws up DNA as well. Because uracil it goes through an enzymatic process where a methyl group's added, becomes, of course, thymine. So you're starving the cells for thymine, that screws up the DNA, and you've got a direct screw up with the RNA. So cells try to replicate, they make lots of damage to RNA and DNA, and then they die. But of course, it does this in every cell in your body. But actually, 5-fluorouracil, if your dose is 100%, less than 1% actually gets associated with the cancer. And of that, first thing that happens, it gets phosphorylated, and 0.5% is effective. I was on it, and I don't want to elaborate too much, but it gave me terrible constipation combined with explosive diarrhea. That had a very bad outcome. That was a joke too, but it actually happened. So they developed this because some very bright person realized, hey, uracil, thymine, adenine, now we have kind of a playground, and we know that cancer divides very rapidly. So in the early 60s, I'm not sure, I guess we had oncologists there, and it was a subset. The oncologist with the FDA said, hey, we can go after cancer with some of these drugs that associate with rapidly dividing cells. But of course, the paradigm for chemotherapy has been target the rapidly dividing cells, try not to kill the patient. And I got involved in the nanomedicine aspect because we can encapsulate the 5-FU, so it's not systemically delivered, and we can target it to the specific cancer because most of the cancer cells have very specific, including triple negative. We use anti-CD71 to target particles to the triple negative breast cancer, and so we can kill it without any systemic effects. And that's going to be interesting because our next animal trial, we're going to go after metastatic breast cancer in bone in an animal with Andrea Mostra. So I don't think it's necessarily, they're going to be new therapies developed. We still want to kill the cancer, but we want to kill the cancer to do no harm to the patient. And that's where the nanoparticles come in. It's a disruptive technology because basically it's like telling to the drug companies, you guys have been getting it wrong for like 60 years. Not strictly true. I survived conventional chemo, and my wife survived conventional chemo. We both personally have aftershocks. We're both type 2 diabetics. Maybe if I lose that 25 pounds, I won't be. But we're both type 2 diabetics, and my wife doesn't need to lose any weight at all. And I've got neuropathy and still zombie hands and kind of zombie feet. So it often works, but pancreatic cancer, 91% of the cases are metastatic when they're discovered. Lung cancer is something like a 19% survival rate at 5 years. That's the #1 killer pretty much throughout the world. And most of those people aren't smokers, actually. You don't have to smoke to get lung cancer. There's a genetic defect, or I'm not sure how to put it. Those of us who, along with people who get cancer without smoking or other environmental factors, when we're squatting by the fires, some of us developed a mutation that enabled us to handle the carcinogen that comes from smoke. Other people didn't. So in a modern world, I doubt very seriously there's more particulate in the air most places than there was back in the 18th century, in the 17th century, when coal was filling the air full of soot. So do I think we need new chemotherapeutics? Yes, I do. But now we can target, now we can encapsulate and literally cause no side effects. And so in the trial we're going to be running this summer, you bet we're going to, it's hard to collect mouse urine, but it's much easier to collect mouse feces. And we're going to be looking at the microbiome in the mouse feces, both with a baseline and healthy, these are going to be athymic mice, so they're not healthy by definition. They're missing the T cells because they were born without a thymus. But we'll be able to look at their microbiome and see if it changes with the free drug versus the encapsulated drug. And that's going to be an interesting study. And then all of our animal trials in the future will incorporate looking at the microbiome because it's there. We know it's important, and therefore we're going to start to collect a lot of data on it. So I didn't really answer your question, but 5FU is a pretty good example of how some bright person said, oh, we can mess with the uracil. And that's often the case. Taxol is a different sort of case. You know the story about Taxol. So someone was looking at, I think someone got the word that somewhere in a third world country, people had a tea they made from the bark of the yew tree. And it seemed to have an effect on cancer in the local population. And so pharma heard about this and and I don't know who the original finder was, but they found that the yew bark had this molecule that they called Taxol. And then actually at the University of Florida, I remember it, one of our medicinal pharmacists or pharmacist chemists developed the first synthetic pathway for Taxol. And the reason I remember it is he and his team got the 4 millionth US patent that had been awarded. That's the only reason I remembered at the time. My wife was on the docetaxel, which is Taxotere, which is Taxol basically. So that was discovered from that natural products. Dr. Amin down at Hershey has discovered an anti-carcinogenic molecule, and you're going to love this, broccoli. Who eats broccoli? I do. I love broccoli. So broccoli has a naturally occurring anti-carcinic, and people are starting to turn toward that. I have a colleague at Brian Barth who was on the papers around the immune effect. Brian is studying different natural products from blackberries and blueberries. Blackberries have long been known to have anti-carcinogenic effects. So eat your blackberries, folks, and maybe blueberries. I prefer blueberries personally. So they're looking at natural products now.
So when Jim says, I have a colleague that studies this and this and this, he's actually the most connected person at Penn State with all of the scientists at Penn State. So he's collaborated with more people than anyone else has. So if you look at a diagram of who's connected, he's like at the center. And so when he says, I have a colleague, he's got a lot of them.
But I only work with people who produce. And they only work with me if I produce. But there's a very simple way. I came in, I mean, I was out of medicine, medical research from 1982 when I had my second child on the way, and I knew I had a job at Patel. Now, I was in Australia. And I went mainstream material science until 2003, when I got the word from Larry Sinoway down in Hershey that my heart disease was unexpectedly completely gone. And so I decided, we had all this nanoscience, and so I decided to look around for more meaningful applications than making ceramic widgets. And I went to a conference in Seoul. I did a lot of traveling in 2003. I went to Seoul, I went to the UK, and Ai-Wei Chin from Penn was talking about using nano silica as a delivery system for drugs, surface delivery, which we know now doesn't work. If you put a chemotherapeutic on the surface of something, or if you put it on the inside of most polymeric systems, as soon as you deploy that in the bloodstream, it begins to degrade and release. So you don't really mitigate most of the side effects. So I came back, I thought about it. I had a wonderful graduate student, Sarah Rouse, now Sarah Rouse Norden, and she was on the NSF fellowship. I said, Sarah, I had this idea of encapsulating fluorophores. Would you like to give it a try? She said, sure, doctor, there. And she had been doing some work on silicon dioxide. We'd been making small 20 nanometer silica for a decade with metal and semiconductor cores and looking at optical properties. And so that was late August of 2003. By Christmas, I had beautifully fluorescent formulations. And I was looking for a collaborator. I'd been out of medicine a long time. I ran into our vice president in charge of IP, who was leaving the job at a Christmas party, actually at Carlo Pantano's house. He was director of the MRI at the time. I said, Garrett, you know anyone down at Hershey? He goes, Mark Kester. And then, of course, I talked to Larry Sinoway. That's why he's on the patent, because he said, you know what we really need for your disease, where the adrenaline receptors are screwed up? We need something we can image as a metric as to whether the adrenal receptors are still active or they're faded into kind of tectonic collapse, which is the end stage for cardiomyopathy or weakened heart. And so we're just getting around to that because we had to develop a photoacoustic system to detect those particles. But so I met with Mark and he got the technology like that. I can't tell you how many pharmaceutical people go, huh? I mean, the vast majority. And because Mark was vested in nanoliposome delivery systems. And so he goes, well, silica won't work because it's not bioresorbable, it's mildly cytotoxic. And it took probably 2 minutes, because I come out of kidney stone literature, say, well, it's got to be calcium phosphate. Well, it turns out during development, we had just a smidgen of silicate to stabilize the amorphous phase. You can't put drugs into crystalline materials because they just don't fit. Whereas amorphous materials, and calcium phosphate starts out as an amorphous material does. So Sarah, that was a lot more difficult development. That took about 9 months. And then we started just having a lot of fun encapsulating stuff. Our encapsulation efficiency was rather poor. But we had highly fluorescent systems. We encapsulated Kester's ceramide, which is actually the ceramide nanoliposomes going through the clinical trial now. But to improve the phosphorylation, or the 5-FU and some of these other drugs, we're working on docetaxel now. we know if we have phosphate groups, we have a huge amount of encapsulation because the phosphate binds to the calcium in our particles. No one in pharmacology or medicine would want to use a phosphated drug though. You put it in the bloodstream and it gets cleared within minutes. Probably got a half-life of 12 minutes. The first time it hits the liver, it's gone, or the spleen. It undergoes phagocytosis and gets cleared. But I explained to our collaborators who blocked that, I said, guys, we're going to put it inside the particle. It's not going to be exposed to the bloodstream. So the 5-FU, we had been struggling with for 2 or 3 years. It absorbed nowhere. It didn't get taken up anywhere. The fluoride's not active, of course, because it only has one bond and it doesn't bridge. And so we looked at that. We were writing a review paper on the metabolic pathway for 5-FU, arguably the oldest drug out there. And we discovered that the first thing that happens to 5-FU when it does go across the cell membrane, it gets a ribose phosphate attached. We tried that. We went from 0.01% uptake, which was laughable, to 30, 40, 50 mole percent. Moles in, moles out. And it's much more lethal because it turns out one of the reasons at 1% that finds the cell membrane, you only have 0.5% effectiveness, is because the 5-FU has 2 places where it gets phosphorylated, and the second place is inactive. So we're picking the active drug, and we get beautiful knockdown at 1,000th of the dosage of the free drug. We're in the nanomolar range, and you have to give the free drug up in almost a millimolar. And 5-FU itself has a very narrow therapeutic window, below which it's ineffective, above which it's lethal. So these drugs, I think, stand good hard looks. Most of the pharmacologists don't like us either. But my attitude is if you're going to do drug discovery, develop something more benign that you can target. Targeting is everything. So IV versus pills. So in breast cancer in particular, they're basically 3 basic flavors. What we target, well, HER2, and you can use an anti-HER2, antisense to target the cell membrane of that cancer. You're going to get breast cancer, and guys do get breast cancer too. The HER2 is the most treatable. And then you have the HER2, you have the, estrogen. That's the most treatable. And you can target the estrogen with the anti-estrogen. But of course, one of the side effects when you're delivering the antisense estrogen is there estrogen all over the female body, including the ovary. And 1% of the patients will get endometriosis induced by the estrogen treatment. This is called tamoxifen. The one for the HER2 is called Herceptin. And that's used for 5 years as oral dosages. And then you have the triple negative like we work with, of which the MDA-MB231 is one of the most aggressive triple negatives. And a biochemist discovered a receptor, the anti-CD71. We looked at the transferrin receptor, and that was not specific enough because transferrin is all over every mammalian cell membrane. But the anti-CD71 was unique to that breast cancer, and we've had a lot of success with that. So the targeting combined with some innovation, even with existing drugs, you can remove the drug from this as a systemic treatment. One of the things we're going to do with the docetaxel, which we're working on right now, it causes massive hair loss. Which, to be honest, in guys, we don't really care. I mean, I shaved my head. And I didn't lose the hair on my head. I lost all the hair on my arms and my legs because of the nerve damage. But every woman that I've talked to who's a breast cancer survivor, including my wife, said the single worst moment is when all the hair fell out because it goes boom like that. It's a matter of like 5 minutes. And they don't have to go through that nonsense. So one of the animal, we'll do a Balb CJ mouse that has hair. The nude mice, of course, don't. We'll evaluate hair loss. So these kinds of side effects it largely should be relegated to the dungeons, not in the mainstream population.
So we have 5 minutes, which may not be enough time for another question. But does somebody else have another question?
This wasn't the focus of the talk, but you said the urine is sterile. Understand why that?
Healthy urine is sterile. The reason it's sterile, well, I'm not sure I know the reason, but basically the kidneys are a continuous filter and a continuous crystallizer studying stone disease. And so the species that can get across the Bowman's capsule in the nephron are generally pretty small. You don't see blood cells cross. Our nanoparticles cross and then cross back in. They're at 20 nanometers. If you begin looking at nanoparticles much above 200, certainly 300, they'll get cleared, but they won't get back into the blood. Same thing with the liver. So the urine is a very important reason why urine needs to be sterile. The number one function of the renal system is to remove the sequestered nitrogen byproducts of protein metabolism. The number one is urea. And if a patient through, mostly, has E. coli travel up through the ureter, the urethra, and into the kidney, the E. coli will live off the urea. When it breaks down urea, it goes to CO2 and ammonia. And the ammonia, CO2 doesn't do much to the pH, but the ammonia causes the pH to become alkaline. And under those conditions, the patient, if you have lots of magnesium and phosphate, and now you have ammonia, will form struvite kidney stones. Magnesium, ammonium, phosphate. And they're called infectious stones because you only see this if pH of the urine goes above 8. pH of a healthy urine should be 6.5. And so there's a very good reason why urine is sterile. Any bacteria will start to gobble up the metabolism of urea, and maybe into creatinine, which is another major nitrogen sequestering element. We call those infectious stones.
So I want to thank Jim for coming out. He serves on the Microbiome Executive Committee, and so we're really happy to have that.
This was fun to dive in finally.
Yeah, great. So thanks for coming out. Next week, the meeting will be in another room. It will be on the Microbiome Center notes that come on Tuesday. So just pay attention to that. Thank you, Gary.